So in this video I'm going to show you how to thaw frozen cells. So I've gone to the nitrogen tank, I've gotten a vial of frozen feeder cells here. This could be any adherent cell type, and the procedure is essentially the same. The first thing you want to do is rapidly thaw this vial in a water bath at 37 degrees. So I'm going to put this in a floaty, or I could just hold it and put it in this water bath. You can also use the heat block that's in the lab for this. It should only take a minute or two to thaw. So you don't want to leave it in here for 5-10 minutes. Keep track of it as you go. So in the hood, I have a flask that I'm going to use to plate my cells. The size of the flask that you're using will depend upon how many cells were frozen back in the aliquot that you're thawing. I have my pipette man, and I have my pipettes to go with it. And I also need media that's appropriate for the cell type of interest. And so that will depend on what kind of cells you're thawing, but you need complete media that is good for the cells that you are thawing. So now I should have the cells thawed. You don't want them to get completely, totally warm. You want to be able to see that there's still a little bit of ice, that it's thawed, but there's still a little bit of ice left in the tube. Because if it gets completely warm, these cells are going to start dying pretty fast after they thaw, because there's DMSO in here, which is toxic to cells at the concentration that it's in. So for this procedure, into a small flask like this, I don't have to pellet the cells or anything, because I'm going to dilute the DMSO out far enough that it's not going to be a problem. The easiest way of doing this is to just open the vial, open the flask, and dump the contents of the vial into the flask. Notice that I didn't touch the vial to the flask when I did that, because you don't know that the outside of this vial is clean. And now I have the cells inside my flask here, and I'm going to dilute them out to 12 mils total. This is about 1 mil already with media. This has to be done slowly, because these cells, having just been thawed, are very sensitive to a rapid change in pressure from adding a lot of media. All at once you'll change the osmotic pressure and you'll burst the cells. So I have 11 mils of media in here, but I'm going to add it very slowly. I'm going to add a little bit. I'm going to sort of shake. I'm going to add a little bit more. I'm going to shake. I don't have to worry about measuring specific volumes at this point, because I know that I have 1 mil in there and I have 11 mils in my pipette, so I'm going to end up with 12 mils total. And it should take you around 60 seconds to do this entire process. If you're going any faster, you're endangering sort of bursting your cells. After the first few minutes, I'm going sort of slowly, continuously down with my pipette and sort of shaking continuously so that I'm making sure that I'm mixing the cells as I go. So I'm just pressing on the down button on the pipette just very, very slightly in order to get this slow pipetting out. And then you just keep letting it go slowly and shaking until all of the media is in the flask. Cap the flask, get rid of the pipette, seal the flask, and then you shake side to side like this to make sure the growth surface, the bottom growth surface, is evenly covered. There aren't any spots on the growth surface that are not covered by media. Label the flask so that you know what it is. I usually label it on the end. And then the flask goes into the incubator. And that is how you thaw cells.